Module 3, Part 2. In the first two topics of the Module 3, Part 1, we looked at synthesis of secretory proteins on ER-bound polyribosomes. In this part, we are going to talk about what else happens to those proteins, how they are modified, and what happens beyond the ER on their next step in the secretory pathway, which is the Golgi apparatus. Here, most of the proteins that are in the ER are secretory proteins that are on their way to somewhere else in the cell. They are on their way to the Golgi, and then they are destined for some other locations in the cell, such as the lysosome, the plasma membranes, or even outside the cell in the extracellular space, because they are destined to be secreted. But there is a group of proteins that actually function in the ER and must remain in the ER and are worth mentioning here. One of the protein is a binding protein, or BIP, and we saw this protein already. It is a chaperone protein, which means it binds to other proteins and assists in some function. In this case, the BIP will aid in folding and multimerring assembly of the proteins. So as proteins are being synthesized and extruded into the ER, the BIP can bind to them and help them fold properly. And if proteins must interact with other protein subunit, then BIP can assist in the formation of protein complexes. We already saw BIP when we talked about the post-translational translocation of secretory proteins in the yeast. We saw that the yeast proteins that are translocated in the ER, that BIP can bind to those proteins as they are being extruded out of the ER lumen and prevent their backsliding out of the translocon. And really important function of BIP is its involvement in the unfolded protein response that we are going to talk about soon. So as the protein is being made and it reaches its destination, it is natural for the protein to fold by itself, depending upon the interaction of the amino acids with the environment in a protein it is in. But several proteins need some chaperones like BIP. Another protein is the peptidyl prolyl isomerase, or PPI. So the peptidyl prolyl isomerase is a very important key enzyme which can interconvert the cis-trans isomer with the proline. Now, proline is an unusual amino acid, conformationally restrained because of the peptide bond due to its cyclic nature, and the side chain is bonded with the secondary amine nitrogen. Now, most peptide bonds are trans bonds and due to the steric hindrance, but the proline's unusual structure can stabilize in the biological form in the cis form, and both the cis and the trans form, which is other one is flipped, can exist in the nature under natural conditions, and is extremely important for protein folding. So the peptidyl prolyl isomerase actually accelerates this rotation about the peptidyl prolyl bonds in the unfolded segments of a polypeptide, and this is very important for the protein folding function. Another important protein in the ER lumen is the protein disulfide isomerase, or PDI, and it catalyzes the formation of disulfide bridges in protein, which also aids in the appropriate folding. So we are going to take a look at these. The PDI forms and rearrange protein cysteine disulfide bonds. Its active sites contains two closely spaced cysteines and easily interconverted between the reduced dithiol form and the oxidized disulfide form. So in the upper panel, what you can see that the substrate proteins have the ionized cysteine thiol reacts with the disulfide bond in oxidized PDI to form a disulfide bonded PDI substrate protein intermediate. So then the substrate protein's second ionized thiol reacts with the intermediate, forming a disulfide bond within the substrate protein and releasing the reduced PDI. The PDI disulfide bond is formed by electron transfer to the reduced to an ERO1, which is also an ER luminal protein and helps in regeneration of the PDI. The red arrows right here shows the electron transfers between the thiols. So that's the formation of disulfide bond in a nascent polypeptide chain. Now the bottom panel shows the rearrangement of improper disulfide bonds. Sometimes the PDI can make mistakes and wrong disulfide bonds can be formed, but the PDI can correct those mistakes by repeated reactions until the protein is folded in the most thermodynamically favorable condition. So very important protein. So here is an example of the influenza virus hemagglutinin folding and assembly. When you talk about the influenza virus and refer to H1, N1, H5, N1, etc., that H refers to this hemagglutinin, which is a membrane-bound glycoprotein as well. This protein is made and properly folded in our cells during a viral infection, and this is inserted onto the envelope of the influenza virus. Now, without going into the much detail, what you can appreciate that how the ER-resident chaperone proteins like BIP, PDI, and glycosyl transferases modify this protein to fold properly. The carbohydrate-binding lectin proteins, on the other hand, prevent the premature folding of the protein. And finally, the hemagglutinin is assembled into the trimeric form, as you can see in the picture. So this is an overview of a protein folding of a complex protein. But sometimes proteins are synthesized incorrectly, or sometimes during their synthesis, they fold incorrectly, or sometimes the cell is making too much of a certain protein. And this triggers what is known as the unfolded protein response. You don't want that unfolded protein, which is likely to be non-functional, to continue on its path in the secretory pathway to the Golgi to its final destination, because this could be fairly detrimental to the cell if it has a mutant receptors on its cell surface, or it has a secreting non-functional hormones, or it is a lysosomal hydrolase, perhaps not folded properly, so it won't work in the context of lysosome. One of the BIP's binding proteins function is to police the folding of these secretory protein. A fraction of the BIP in the ER is associated with an endonuclease transmembrane protein located in the ER known as an IRE1. Now, when BIP binds to the IRE1, it keeps the IRE1 monomeric state or as a single monomer of IRE1. But if the unfolded proteins occur in the lumen of the ER, BIP will bind to these proteins and it will come off the IRE1 endonuclease. And in that case, when the IRE1 is empty, they form the dimerization. So this is the dimerization of the IRE1, which is the active form. And these endonucleases are activated in this point. Now, in the cytosol of the cell, the messenger RNA that encodes a transcription factor known as HAC1, and in this example, it is unspliced. When IRE1 endonuclease become activated, it can bind to the HAC1 messenger RNA and cleaves it in such a way that now you have a mature functional RNA encoding this transcription factor. The transcription factor becomes translated, it moves to the nucleus, and it regulates the expression of genes that are involved in the unfolded protein response. Additionally, these unfolded proteins are moved out of the ER and degraded by the cytoplasmic proteases or by the proteasome pathways. Now you will see I have a question mark here because the molecular mechanism by which these proteins get transported out of the ER into the cytosol and what regulates that is currently unknown. So this is an early quality control event, and we will discuss another quality control event later in the sequence. Now finally, there are glycosyl transferases and which glycosylate the proteins in the ER. There is a large collection of these in the ER, and in fact, we could like to focus on these enzymes now in particular for the ER function in glycosylating the secretory protein. So the most important function in the endoplasmic reticulum is to glycosylate the secretory proteins. And I would like to say that the majority of the secretory proteins are glycosylated, or it might be safe to say that every single protein is glycosylated. A lot of progress was made in understanding this process when it was discovered that sugars or saccharides are added to these secretory proteins in large groups. So there's a large preformed oligosaccharide complex, and it is transferred en masse in total to the secretory proteins. One of the places you will find these oligosaccharide modification on these secretory proteins are on the asparagine residues. And not any asparagine residue and not every asparagine residue, but asparagine residues that are usually in this sequence combination. So you have an asparagine and you have an X, which is any protein, any amino acid in the protein sequence, followed by a serine or threonine. The asparagine residue which are found in this context are usually targeted for sugar modification. The type of sugar modification that I'm talking about here is specifically what is called as an N-linked, right here, because this is an amino group, N-linked oligosaccharides. And again, just repeating, they are so named because the single amino acid code for asparagine is N. The N-asparagine is the link to the oligosaccharides, or the modification is added to these asparagine residues. The initial step is that a very large oligosaccharide complex is formed, and we are going to see that in a second, that how this oligosaccharide is being formed in a minute. But it's a very large complex, and it is made up of glucose, mannose, and N-acetylglucosamine. And we are going to see that how this is actually formed. So here, let's first take a look at the biosynthesis of this N-linked oligosaccharide precursors. The oligosaccharide precursors containing 14 residues preformed on the dolichol phosphate, which is a lipid embedded in the ER membrane. And this dolichol phosphate is a hydrophobic, it's a polyisoprenoid, and it contains about 75 to 95 carbon atoms and docked into the membrane. Now, what happens, the first N-acetylglucosamine is bound by the nucleotide sugar, UDP, which is the uridyl diphosphate. So this is a pentose sugar with a uridine, two phosphates, and to 2-phosphate, there is the sugar attached to it. And the first reaction is a high-energy pyrophosphate link, which is added on top of the phosphate that the dolichol phosphate already have, and that will first attach the first N-acetylglucosamine. And this is a step that I have written that is blocked by tunicamycin. So it's very important. So now this will be followed by addition of several others. So there will be a second UDP coming in, adding the second, followed by the addition of the 5 mannose residues, and each time this will be brought by a GDP. Once these 7 residues are added to the dolichol phosphate, it flips into the membrane, and then additional mannose residues are delivered to the dolichol phosphate. So you can easily see that here, right here, this sugar where the GDP is adding the mannose right here, it will flip back, transfer that mannose right here, and then the empty dolichol phosphate will flip back and again will bring the next one. And that's how the mannose chain goes on. And then finally, what happens, that the glucose is added to these, and 3 glucose residues, and these are added by the UDP as well, uridyl diphosphate, and with this flipping mechanism as well. The dolichol phosphate binds to 1 glucose, which flips back and delivers the glucose until there are 3 of them. So here is what the big picture looks like. You have a secretory protein being extruded into the lumen of the ER. Here is the asparagine residue, and that is the target for the N-glycosylation. Here you can see that this is a very large oligosaccharide complex that is made up of glucose, mannose, and N-acetylglucosamine, and is lipid anchored with the dolichol phosphate right here. And so the complex is transferred en bloc or en masse, this whole thing, into the asparagine residue as it becomes available for modification and becomes extruded into the lumen of the ER. So that's the first step of adding the N-linked oligosaccharide to these secretory proteins. Also in the ER, you can see a little bit of trimming. What we mean by a trimming, and so once a large sugar complex is added to these proteins, you get trimming of these sugars as shown here. You can see that there are 3 glucose and 1 mannose are removed from this complex. So this is the slide, is just another rendition of the stepwise trimming of the sugar residues as the proteins are getting prepared to be transported towards the cis Golgi by vesicular transport. This is the same as the last slide where there are 3 glucose and mannose is trimmed out of the matured ER protein to be destined to Golgi apparatus. So in the next step for this protein is to move from the endoplasmic reticulum to the Golgi as shown here. So how do these vesicles move from ER to the Golgi? All of these compartments along the secretory pathway communicate with each other through vesicle trafficking. As the name suggests, the vesicle trafficking is a process by which a vesicle buds off from a compartment, which is called a donor compartment, and it fuses with the target compartment. You can imagine that it is a highly regulated process. As this vesicle bud from the donor compartment, it has to try to only pick up the protein cargo that is destined for movement along the secretory pathway. It must fuse with the correct target compartment. So a vesicle that buds from the ER has to fuse with the Golgi. So how does the cell know it has to fuse with the Golgi, and how does the cell prevent the vesicle from fusing with lysosomes or other organelle in the cell? In the last part of this topic, we'll talk about the molecular mechanism that regulates the specificity of this vesicle trafficking event. So here is a drawing of a Golgi apparatus. As you have seen before, it has the cis face and a trans face. It is the cis side of the organelle that vesicles from the ER fuse, and there is a series of flattened sacs of membrane in the middle known as cisterna. Each of these cisterna have a specific function in the processing of these secretory proteins. Then, of course, the cisterna that are closest to the cis face right here are called the cis cisterna, and you have a medial cisterna, and then you have a trans cisterna. When you look at the transmission EM of the Golgi apparatus, you can easily see these swarms of vesicles, and these vesicles are fusing to form the Golgi from the ER, leaving the Golgi to deliver the protein cargo to the other parts of the cell. Or you might be seeing vesicle trafficking between the cisterna as well. So here is a transmission EM of the Golgi apparatus from green algae, which is called Chlamydomonas. You can see that very clearly the well-developed flattened stacks of these vesicles right here. And this is the cis face of the Golgi, which is the trans face of the Golgi. And you can see that these are the vesicle which buds off the cisterna and move on to the next one into different stacks from cis to trans face. So we are talking about the N-linked oligosaccharide, and these are sugar modifications on asparagine residues on these secretory proteins. There are actually two classes of N-linked oligosaccharides. Let's start with the second one, and they are called the high mannose N-linked oligosaccharide. As their name suggests, they are high in mannose and shown in blue, and they are formed in the same initial steps for addition of that very large oligosaccharide complex. When you get some trimming in the ER and some additional trimming in the Golgi apparatus, and you end up with this high mannose sugar. Now, we are not going to consider these too much more in the course. The one that we are really going to look into is this complex N-linked oligosaccharide modification on secretory proteins. The way these are formed is by trimming in the ER and then trimming in the Golgi apparatus, and then some additional sugars or addition of some more sugars such as galactose and sialic acid. So let's take a look at the complex N-linked oligosaccharide. So here we are back in the ER. This is a repeat of the initial step that we have seen before. So you have this asparagine residues and this complex of sugar added as one whole preformed complex, what we say as N-block to this asparagine. Then you have an enzyme, glucosidase 1, right here, which removes one glucose residue. You have a second enzyme, which is known as a glucosidase 2, which removes two additional glucose residues. And then you have a mannosidase that removes a mannose residue while in the ER. So here, these are all ER resident proteins And these are known as the ER glucosidase 1, ER glucosidase 2, ER mannose, and sometimes they are also known as the alpha glucosidase 2, alpha mannose, because they cleave at the alpha carbon to remove these residues. So here, the initial 9 mannose is reduced to 8 mannose in the final product, and at that point, the protein is destined to the Golgi, but not yet. There is another quality control step before the protein can move on to the Golgi. So the misfolded secretory proteins are recognized by the specific ER membrane proteins and targeted for transport from the ER lumen to the cytosol for degradation, as we have already seen before, this through the BIP-IRA response. In addition to other proteins, calnexin and calculin CRT, so this calnexin and the calreticulin bind to the N-linked oligosaccharides, and then in which the glucose is re-added to form an original untrimmed version of the glycosylation as the first step in the sequence, and this promotes the retention of the protein into the ER without transportation into the Golgi. And this aids in the folding by the chaperone activities. Until the protein is properly folded, this process can be repeated over and over. So if the protein failed to fold properly even after this process, they are retained in the ER for longer times and then eventually undergo a mannose trimming by an ER alpha mannosidase. Please note that the previous slide showing the trimming of one of the mannose by the mannosidase from 9 to 8, but here the ER alpha mannosidase removes 4 mannose, so it is reduced to 5 mannose from 9. The unusual trimming pattern promotes the binding of OS9, and this is a protein which dislocates this unfolded protein or misfolded protein out of the ER, followed by ubiquitinylation and degradation by proteasomes. By a mechanism, how this is transferred is not clearly known, that we have seen before. So here are the steps in the Golgi. So we have seen the steps in the modification of these in the ER. Now we are looking at the Golgi. Once this protein enters the Golgi apparatus, you have the removal of 3 additional mannose by this Golgi mannosidase 1. Now then you get the addition of an N-acetylglucosamine and with the enzyme glucosamine transferase, and you have the removal of 2 additional mannoses, as you can see here, with the Golgi mannosidase 2. And then you finally get the addition of 2 N-acetylglucosamine and 3 galactose residues and 3 sialic acid, which is not shown. And you end up with a complex N-linked oligosaccharide modification on this asparagine residue. So this is the overall process which happens in the Golgi. So this dotted line just shows that the middle portion of the Golgi apparatus. So what is really interesting is that all these biochemical reactions, all of these removal of sugars and the addition of sugars are compartmentalized in the Golgi. So only certain reactions happen in certain Golgi stacks. So you have the removal of mannose in the cis-cisterna or the cis stack. In addition of some sugars in the medial stack, in addition of some of the sialic acids and in the very trans region of the Golgi. So this really gives you an idea why the Golgi is structured the way it is. It is these stacks in the Golgi that actually make sure that these biochemical reactions are compartmentalized, and it also makes sure that the sugars are removed and added in the correct order. Because you have to have the removal of some sugars before you have the addition of other sugars, and in fact that is happening in separate stacks or in these separate compartments of Golgi, ensures that you have the correct processing of these N-linked glycans or oligosaccharides. So, is there any way to track the glycosylation of the secretory proteins? In fact, there is a way biochemically using enzymes known as endoglycosidases, or in short, we are called these as endos for short. What these endoglycosidases do is that they can cleave these sugars off these secretory proteins once they are added. So you have this N-linked glycans attached to the asparagine residue, and the endoglycosidases can cut it from and completely cleave it from the asparagine. Something very interesting happens in this step here along the processing and is indicated by the double dotted line here within the medial Golgi. When secretory proteins pass through the medial Golgi and they are processed by the enzyme in the medial Golgi, their sensitivity to these endoglycosidases change. So in this case of endoglycosidase H or endoglycosidase D. So what happens in this case of endoglycosidase H or endo-H, these prematurely processed secretory proteins are endo-H sensitive until they cross this point in the medial Golgi, and then they become resistant to the action of endoglycosidase H. The opposite is true for the endoglycosidase D or the endo-D. These proteins are resistant to the action of endoglycosidase D until the proteins cross this step into the medial Golgi, and then the glycans that are attached to them become sensitive to the endo-H. The exact moment that this happens is when this mannose residue is removed, the second mannose residue is removed. That is when you gain or lose the sensitivity to these particular enzymes. So here you are asking two research questions. First, what happens to the misfolded proteins which retain in the ER? And what happens to the proteins that move into the Golgi and move through the Golgi stacks? So let's first take a look at the panel A on the left. Our first hypothesis is the misfolded protein will be retained in the ER, whereas the properly folded protein will be going out of the ER to the Golgi. And our second hypothesis is that the glycans on the proteins are going to be trimmed as it moves through the Golgi stacks and will become sensitive to endo-D. So this hypothesis is illustrated right here on the left. As you can see from the left panel of the figure A, the protein is misfolded, it is retained in the ER, and the Golgi-specific trimming of the glycans does not happen here. On the right-hand side of the figure A, as the protein is moved through the Golgi stacks, there will be a Golgi-specific trimming of the 3 mannose residues. As you can see right here, you can see that there is removal of this mannose, and right here, this mannose and these two. So you can see that the unfolded protein will now have the endo-D undigested glycans attached into the protein, which will have a higher molecular weight that is retained in the ER, whereas the one that moved through the Golgi with the modification can be digested with the endo-D will have a lower molecular weight. So let's first talk about the protocol of the experimental process, how it is really done in real life. In this experiment, a viral membrane glycoprotein is being studied, which is folded, glycosylated the same way as any other secretory protein will. It is a membrane glycoprotein of the vesicular stomatitis virus, an animal virus, or VSVG in short. The interesting part is that the researchers used a temperature-sensitive mutant of the VSV called TS045. This TS045 VSV mutant grown at 40 degrees will not transport proteins out of the ER, but at 32 degrees, the protein will be transported to the Golgi. The cell used in this experiment was CHO cells, or Chinese hamster ovary cells, which were infected in the culture with the TS045 mutant VSV and pulse labeled with radioactive 35S methionine. To prove the hypothesis experimentally, the cells were split into two groups. Group 1 is the control cells maintained at 40 degrees Celsius, which will have the misfolded proteins were retained in the ER. This is called a non-permissive temperature. Whereas the group 2 will have the experimental cells shifted to 32 degrees Celsius, and the protein moves through the secretory pathway, which is this. So in this experiment, the VSV infected and 35S methionine labeled CHO cells were harvested at different time points and immediately added with ice cold buffer and put on ice to stop vesicle trafficking of the proteins, and then the cells were homogenized and the ER and the Golgi was purified as described before by several sucrose density gradient centrifugation, and they're reconstituted as microsomes by incubating with clarified cytosol and ATP, which helps them to again reconstitute in proper microsomes. The proteins were extracted from the ER and the Golgi microsomes and purified by immunoprecipitation. In short, the VSV-G specific antibodies are added to the protein mixture, which will specifically bind to the VSV-G proteins, which already do have the radioactive labels. Then you add the Staphylococcus protein A attached to some saccharose beads. This is just like a different material, like an agarose beads to the mixture. Remember that the Staphylococcus protein A will bind to the antibody, and a short centrifugation will precipitate the beads, and it will pull down precipitate the beads, and it will pull down the attached antibody and the protein of our interest, which is the VSV-G. And all the unbound proteins will be in the supernatant. And you take off the supernatant, and then you repeat these step a couple of times, wash a couple of times, and other non-specific proteins not bound to the antibody and the protein saccharose will be out. So there will be no background. Now you have your purified protein from the different time point, which is digested with the endo-H. So now what we are doing, that after isolating the process right here, we are going to the panel B. You run an SDS polyacrylamide gel electrophoresis of the digestion mixture, and then the movement is measured by autoradiography, which only shows the VSV-G proteins in the gel purified by the mentioned immunoprecipitation. So here at the 0 minutes, you can only see a one band because it is resistant to endo-H. At 10 minutes, now you can see about 2 bands. They are very thick bands and run very close to each other, but there are definitely 2 bands. You can see the original paper, it particularly shows 2 bands. So in this case, the protein probably crossed the medial Golgi somewhere between 5 to 10 minutes and having the trimmed glycans, and it has become sensitive to the endo-D and having the lower molecular weight compared to the untrimmed version resistant to endo-D. So that's how long it takes to synthesize the protein to maturity. And it's an amazing experimental proof of this hypothesis. The panel C shows the percentage of the endo-D sensitive VSV protein over time derived from this electrophoretic data above. And you just scan and quantify the bands on the autoradiograph and plot it against. And at 40 degrees Celsius, you can see there is almost no increase in the endo-H sensitive protein as the misfolded protein at this non-permissive temperature remained in the ER. But at 32 degrees Celsius, most protein moved from the ER to the cis Golgi, and most of the proteins were modified approximately about 30 to 40 minute range. And that's why it is plateauing off. So here is the same experiment using the endoglycosidase H, but remember, the opposite happens in this case. For endoglycosidase H, the protein is sensitive until it crosses the medial Golgi, and then it becomes resistant. So here is in this example, say at 15 minutes, your protein is still sensitive to endoglycosidase H. So at 15 minutes, the left lane shows the size of your proteins in the absence of endoglycosidase H. On the right lane, the band has traveled farther on your gel in the presence of endoglycosidase H. It is smaller, so it suggests that the endoglycosidase H was able to remove the glycan structure that were attached to it, and it reduced its molecular weight. So it's traveled farther on the gel. So at 30 minutes, what you can see, the protein is still sensitive to endoglycosidase H, but by 45 minutes, your protein is resistant to the endoglycosidase H, and it no longer can cleave these glycans, and no change in the size of the molecular weight happens. And of course, at 60 minutes, there is no change or the size of the molecular weight. And this is how you can see that with a fair amount of confidence that the protein traveled through the medial Golgi somewhere in between 30 to 45 minutes. What is interesting that you can also use the endoglycosidase H to distinguish in between the complex, which is N-linked glycans, and high mannose or complex N-linked sugars. The reason for that is that although the complex N-linked glycans gain resistance to the action of endo-H at the point they cross the medial Golgi, so high mannose modification sugars are always sensitive to endo-H. So say you have isolated a mature protein, a protein which has completed its processing and maybe has been secreted from the cell. Next, you can treat the protein with endo-H. After running on a gel, you can see that this protein is sensitive to the action of endo-H. As the treated protein runs lower on the gel, then no treatment control. If this protein is not sensitive to the action of endo-H, you can conclude that the N-linked glycans on these proteins are not the high mannose type. The glycans on this protein are likely to be the complex N-linked glycan type. Now, this is a very simplified example. Sometimes these secretory proteins have both high mannose and complex N-linked sugars in different amino acid asparagine residues. But I think you can appreciate that from this experiment that how powerful this endo-H or the endo-D is in the study of these protein trafficking. You can also track the secretory protein trafficking by microscopy. As you know from the previous slide, the VSV-G is a protein that traffics to the plasma membrane. Now, these scientists also use the same temperature-sensitive mutant of the VSV-G. Now, they did not use the live virus as the previous experiment. They use a cloned chimeric version of this G protein. Now again, just to repeat, that the cells at 40 degrees Celsius, then the trafficking would be halted and the VSV-G will be retained in the ER. This is the TH045 VSV-G mutant proteins character. So there is a traffic defect at 40 degrees, but just as you have seen before. So what happened is that they express the GFP VSV-G in a mammalian cell as a chimeric protein that we have talked before. So this VSV is now can be tracked as a fluorescent tag is attached to that. They kept the cells at 40 degrees so all the cell GFP VSV would end up into the ER and stay in the ER. So this actually synchronizes the synthesis of this protein. Then you relieve the block. You move the cells into a permissive temperature like the 32 degrees, and the synthesis continues at this point. So you can see at 0 minutes, all of the VSV-G is in the ER right here. 0 minute, all of them is at the ER. By about 40 minutes, it is all in the Golgi, and about 180 minutes, it is all in the plasma membrane of the cell. You can actually take fluorescence measurement and can track them as shown here. You will notice as the GFP VSV-G in the plasma membrane is going down after 180 minutes, and you may be wondering why. It has been suggested that photobleaching is going on. It is not like the cell is losing the GFP, it's subject to photobleaching, so that's normal. So that is why you are likely seeing the decline in the fluorescence. This is really a cool experiment because you can actually do fluorescence video microscopy to track in real time and watch this happen. The disadvantage is that the resolution is not as fine as you have used in endoglycosidases. You really cannot say in this particular example when the protein crosses the medial Golgi stacks or medial Golgi cisterna. The only conclusion that you can make at 40 minutes that this protein finds itself in the Golgi, and beyond that, it is the plasma membrane, but you cannot actually discern when the protein moves from stack to stack. So this is one of the disadvantages. The advantage is that you can see this with a microscope and it is in real time and it is extremely impressive. So there are 4 short additional points we want to make about the secretory protein trafficking. We want to talk about the ER resident proteins again. We want to stress that the N-glycans are not the only sugar modifications. We want to talk about why it is important to think about the topology of glycoproteins in the membranes. We talked about this a little bit before. And we want to talk about the role of glycosylation. So we discussed that there are some resident proteins that must stay in the ER because they function in the ER. So we are talking about the BIP, or the binding protein. We are talking about the protein disulfide isomerases, protein prolyl isomerases, which is the PPI, and the glucosyl transferases that are responsible for putting those sugar residues on the asparagine amino acid, and also some of those glucosidases and mannosidases that are responsible for some initial trimming of the N-linked glycans in the ER. So they need to stay in the ER. They don't need to move on to the Golgi. So how do the cell control that? So as it turns out, most of these ER resident proteins have a specific sequence on their C-terminus that is made up of with this lysine, aspartic acid, glutamic acid, and leucine. This C-terminus is sometimes called as the KDEL for the single amino acid codes for the lysine, aspartic acid, glutamic acid, and the leucine. And it is this KDEL sequence that tells the cell that these proteins belong to the ER. So what happens when a vesicle buds? So here is the ER right here on the bottom of the slide. You have some soluble secretory proteins that are being made. You see them in the blue. They need to move onto the Golgi apparatus. And then you have these ER resident proteins here that are green. And you can see that they have the KDEL sequence at their C-terminus. Occasionally, these vesicles bud from the ER, and ER resident proteins actually gets caught up in these vesicles, and the vesicles move to the Golgi. So these ER resident proteins end up in the cis-Golgi network. So the cells need some way to retrieve those back into the ER. As it turns out, on the membrane of the ER and to the extension of the membrane of the Golgi apparatus on the vesicles that are trafficking, you have what are known as KDEL receptors here, which are transmembrane proteins that have specificity for the KDEL sequence. So what happens is if a KDEL protein is accidentally caught up in these transport vesicle that belongs in the ER, they can be bound to these KDEL sequences right here on the receptor, and there's a specific retrograde trafficking in which the vesicles move from the Golgi back into the ER. And it is this pathway that these ER resident proteins bind through their KDEL sequences to the KDEL receptors and are returned to the endoplasmic reticulum. So this cell has a safety net that in the event that the ER resident protein is accidentally transported to the Golgi, it can be retrieved back into the ER. So the second point we want to discuss is that the N-linked glycans are not only glycans. There is another class of glycans. We are not going to go into consider that in the course, but it's important that you know that they exist. These are called O-linked oligosaccharides. They are so named because they are bound to the OH groups of the serines and the threonines on these secretory proteins. And here are some examples. An O-linked oligosaccharides, classic example is a collagen and glycophorin. And collagen is an abundant protein in your connective tissue. So the third point that we want to make is about the topology of the oligosaccharides on the glycoproteins. We have talked about this earlier though, and we just want to make the point we are trying to make here is that ER lumen is equivalent to the extracellular space. So the part of the secretory protein that is found in the ER lumen and is trafficked to the plasma membrane, it will end up on the extracellular side of the cell. So let's see how it happens. So here is in the illustration, in the transmembrane protein, these squiggles right here are being made through the rough endoplasmic reticulum, and the red dots are some kind of glycosylation, say an N-linked glycosylation or O-linked oligosaccharides or both. So you can see the N-linked glycosylation is in the ER lumen. The orientation is preserved in the vesicles and is preserved when the vesicle fuses to the Golgi apparatus. So when the vesicle buds from the ER as shown here, the N-linked glycan or the oligosaccharide is still in the lumen of that vesicle. And when that vesicle fuses with the Golgi apparatus, the N-linked glycan is still in the lumen of the Golgi apparatus. This makes sense because these N-linked glycans needed to be further processed by the Golgi apparatus. So now over here, that the vesicle that is budding from the Golgi apparatus, again, the end-linked portions are in the lumen of the transporter vesicle, and now the transport vesicle moves to the plasma membrane and fuses with the plasma membrane. And at that point, the end-linked glycan is on the outside of the cell. So the lumen of the ER, the lumen of these transport vesicles, and the lumen of the Golgi is equivalent to the extracellular space of the cell. So the last point we want to make is that why the glycosylate these proteins at all? It is not really known why these proteins are glycosylated for sure, but it is hypothesized that the glycosylation participates in folding of the proteins. For example, these large sugar modification on these proteins can control or regulate the way these proteins fold, which can be important in their function. Do they aid in transport? Only very rarely signals to the transporters in different parts of the cell, except in the case of lysosomal targeting. Their sugar modifications that we'll talk about in the future topic actually helps these glycoproteins get into the right organelle, which is the lysosome. They provide stability to these proteins. You can imagine that a secretory protein that has a lot of these end-linked glycans on them, the polypeptide chain would be hidden from the proteases. So they become resistant to proteases. And there is some evidence that some of these proteins are found in the membrane of the lysosome. The lysosome is filled with the proteases, and it is the end-linked glycans or all of these sugar modifications that protect them from the hydrolysis by these proteins. Then it is also thought that the proteins, especially those found in multi-complexes, rely on their glycans for appropriate interactions with other proteins. This concludes Module 3, Part 2.